It's in the nose. So we're going to draw a big-ass nose. Here's the big-ass nose. Not the entire thing, it's inside. I know that it's really, really weird. So this is the top, the roof of your nasal cavity. So nostrils are somewhere here. I'm not drawing them, okay? That makes sense? Here, in this top, you have special cells called olfactory neurons. Okay, so these are, the blue ones are olfactory neurons. When you look at these olfactory neurons, can you see different parts of a neuron? Can you see the body? Can you see the axon? Can you see the dendrites? So these dendrites of olfactory neurons have kind of a specific name. They're called olfactory cilia. These are the only cilia in the human organism that do not move by themselves. They do not move. It's not the intention. Are we clear about that? So, what is the function of dendrites, in this case olfactory cilia? Do they receive the information or do they transmit? They receive, right? So, if you have, and what is smell? Smell is a chemical, okay? So you have a chemical coming in. This chemical gets dissolved in the mucus. And this is really important. Your sense of smell will be significantly impaired when your nasal cavity is overly dry. Am I clear? Because you have nothing to dissolve chemicals in. So now, check this out. Chemical is dissolved in the mucus, and receptors in the olfactory cilia, okay, they sense the chemical, they form some kind of action potential, talk about it, okay, and gradient potential, whatever. And that electrical signal goes through the body into the axon. Axons here, called filaments of olfactory nerves. Sometimes they're called olfactory filaments, you know, that kind of stuff. Does it make sense? And then your olfactory neurons, synapse, with so-called mitral cells. In the structures, they're called glomeruli, or glomerulus is the single. So glomerulus. You have 3 glomeruli here on this image. Are we clear? We're good? So again, first of all, if you need to sense the smell that is really, really faint, what you're going to do? You're going to sniff, right? Why? Because you can push the air into the roof of your nasal cavity, where your olfactory epithelium is. This chemical is going to get dissolved in the mucus. It's going to interact with the receptors on the olfactory cilia, form the signal will be transmitted through the axon, filaments of olfactory nerve, to the mitral cells and the synapse in the glomeruli. Last little bit, sort of anatomical, before we move on to other stuff. This part here, this bow, the roof of the nasal cavity, is formed by cribriform plate of the ethmoid bone. Okay. Now this is something, I think I mentioned that before, did I not? About the, oh yeah, we were talking about olfactory nerve. Look, this is basically a passage to your brain. This is the brain. Are you following? This is why intranasal application of drugs is so effective, like, you know, drugs that have really strong sort of neurological effect, like cocaine. When it is snorted, it gets straight into the brain. That makes sense? Okay. So, we are not the greatest animals in terms of olfaction. For a couple of reasons. First of all, we don't distinguish many smells. And second, we're not super sensitive. We're not super sensitive for one simple reason. Our mitral cells have really high threshold of activation. So you need a lot of stimulus right here to activate them. But let's talk about how we can sense, actually, a significant number of chemicals. It is estimated that human nose can distinguish between approximately 10,000 different smells. Are you with me? Does that make sense? So, if we would have 10,000 different receptors, we wouldn't have space in our genome for anything else. Right? We have 20,000 genes. Well, 25, give or take. Okay? If we spend half of that capacity just for smell, that's a waste of space, enormous. So, how can we get around this? And the mechanism is just incredibly elegant. So imagine, I'm going to draw a schematic. Imagine that you have three receptors and three odorants. Okay? So I'm going to have receptor 1, receptor 2, and receptor 3. And I'm going to have odorant 1, odorant 2, and odorant 3. Now, please, here's the thing. These odorants are somewhat structurally similar. Are you following? They have certain structural things in common, okay? So, turns out that receptor 1 interacts with odorant 1 extremely strong. We're going to give it a score of 5, okay? Odorant 2, nah, kind of meh. And odorant 3, kind of lukewarm. Okay? What about receptor 2? Turns out that receptor 2 interacts with odorants like this. 4, 5, and 1. Okay? And those numbers are completely bogus. This is just for illustrative purposes. So you see that pattern, right? So here's the deal. Each receptor on your olfactory neurons can interact with multiple odorants. Does that statement make sense? Each odorant can interact differently with different receptors. Are you with me? So, this is the question, like it's a hard question. It's a question like 10 out of 10, it's associative thinking. The system of sensing smells has a very specific name, which matches the name of one musical instrument. And you have, like, when one odorant presses the buttons of multiple receptors but different. It's called an accordion system. It is really it is an accordion system. It was discovered in, Nobel Prize was given in 2004 to Linda Buchan, some dude. Okay, but the point is, so one receptor, many odorants. One odorant, many receptors. This is what I illustrated here. Does that make sense? Now I'm going to tell you one really, really important thing. No, it was important too. That thing that I'm going to tell you is also important. Okay? Here's the thing. One neuron carries only one type of receptors. Does that make sense? So for instance, this receptor, this neuron may carry only receptor 1, this only receptor 2. One neuron cannot carry different receptors. And also, all olfactory neurons in the glomerulus carry the same receptor. Does that make sense? Good. Moving on. So, last couple of things to chat about: molecular mechanism of the responses and what happens in your brain. Okay? So, molecular mechanisms. Again, it's going to be second messenger system. So here's your membrane. Receptor. Okay. So here's your odorant. Check this out. Here's your odorant that binds to the receptor. All familiar things. G protein gets activated. Okay? Binds to another enzyme and activates it. This enzyme is called denylate cyclase. Okay? What it does? Well, it takes ATP and converts it into a signaling molecule, second messenger molecule, called cyclic AMP. And what cyclic AMP does? It opens cation channel and when it opens that, what happens to sodium and calcium? They diffuse in. That makes sense? And that results in depolarization. That's all. Much less steps. Questions on this one? We're going to see similar one with the taste. Okay, good. Last little bit. Any questions on this one? Okay, last little bit on the smell. Brain structures that I want you to kind of, I'm going to highlight some structures. So we don't have that high capacity for the smell recognition, but there's a couple of structures that are super important. So hypothalamus, physiological response to the smell. Okay. It's kind of an interesting thing, different, can you imagine a smell that will make you barf? Probably there is, right? That's it. We don't really know why you are barfing at this smell, but there are sort of universal responses, sort of, like rotting flesh, basically. Amygdala, emotional responses to the smell. And hippocampus, memory of the smell. It's kind of bringing us back to the okay? Questions on this one? Any questions on the smell? Everything clear?
Yeah.
Let me know if you can't see something.
Yeah,
Because
I know. What's the memory? Is it formation?
yeah, memory formation. I know, chicken scratch. Good?